Not speaking in French, and my thanks goes to the translators working hard up the back there for us. And thank you, my gratitude goes to Jean-Pierre and the organisers for giving me this opportunity to speak with you all today and to share our experiences and knowledge, because it's an incredible opportunity to do so. So I'm from the Kirby Institute at the University of New South Wales in Sydney, and our program is the Viral Hepatitis and Clinical Research Program. We coordinate a lot of investigator-led trials looking at the scale-up of DAA therapy in prisons and among HIV-positive men who have sex with men, for example. Also cohort studies looking at the comprehensive intervention, prevention strategies and tools. But today I'll be speaking more in line with the simplified diagnostics that might aid the simplification strategies that were just discussed. So today I'll talk about what are the gaps that remain in HCV care, what new diagnostics tools that exist that may help close those gaps, which simplified strategies may help each of the specific communities that we're talking about, as no diagnostic or treatment strategy is going to work for all, as we understand, and what are the steps in implementing these. So this is a slide that you're all well familiar with, discussing 100% of those infected all the way down through to the small proportion of people that actually receive treatment and achieve cure. And we've just heard about the wonderful opportunity that we have with this highly effective treatment coined as Perfectavir. But what are we able to do about all the remaining barriers that do exist prior to receiving this treatment? So one tool that may help close some of these gaps is the development of simple and affordable tests specifically to diagnose active infection. So what tools do exist? So currently HCV, as you know, is diagnosed by venipuncture routinely with an antibody HCV test, followed by confirmation of infection with a quantitative RNA test, which can then determine whether the patient has spontaneously cleared the virus, responded to treatment, is chronic, or, as we've been discussing, is suffering from either relapse from therapy or a reinfection. So obviously antibody tests do have limitations in that they're unable to distinguish between active and resolved infection. They're also unreliable in determining the early acute cases of infection, and sometimes they're also impaired in HIV-positive individuals. But likewise, the quantitative RNA tests that exist have limitations in that they're expensive, although these prices are coming down rapidly. The testing requires careful sample handling, complex equipment and training, and is prone to contamination. So what new diagnostic tools do we have that may help close these gaps? So one avenue that holds a lot of promise for HCV testing is the use of dried blood spots. So these dried blood spots are an alternative form of sample collection, and it's just a simple finger prick where the drops of blood are dried onto filter paper. It's dried overnight or for a couple of hours, and then considered non-infectious at that stage, they can be posted in the mail to a central lab. So the advantages here are obvious in that it's a painless and less non-invasive method, requires significantly less medical training to collect the sample, far simpler transport costs, as it can just go in the internal mail, and there's evidence to show that this sample collection does help access high-risk populations. So rapid point-of-care tests have been discussed, particularly in a session that we spoke of yesterday, and the majority of these that are FDA approved as well are referring to lateral flow antibody tests. But I'm going to discuss the development more with regards to point-of-care tests for the detection and quantitation of RNA. So an example of that would be, there are several in development, but an example of that one is approved is a Cepheid Gene Expert, and it's a cartridge-based system which you put the plasma in, it amplifies and gives you a result in less than 90 minutes. So the advantages here, obviously, are increased cost effectiveness overall on a public health scale if you take into account all the follow-up testings and loss. It's suitable for primary care facilities, as we know, might be more amenable to the HCV affected communities. They have portable versions of these, so therefore applicable to remote settings. But more importantly, it reduced the test to treat cycle, so a patient can come in for one test, receive treatment in that single visit. So therefore, you have an improved linkage to care and patient satisfaction with a test. There are disadvantages, however, of course. They are higher cost per test, so often it's difficult to assess the economics of implementation and the advantages of this. They have quality assurance and regulatory issues that are different to centralised testing. Again, data management and connectivity needs to be managed. Sensitivity of the assay obviously needs to be assessed, and likewise the device performance in the setting that it's being conducted. And something that needs to be addressed also is the user and provider perceptions, and often it's considered that, is a point of care test really as good as a laboratory test? So the core antigen assay is another option that was developed in the early 1990s but has received a lot of more attention in more recent years. And it is a chemiluminescent assay that detects the nucleocapsid protein and allows detection of active infection. The Architect platform that is currently the standard test for core antigen is highly automated. The advantages here is that it detects active infection, it's easy to perform, it is less expensive than an RNA test, and the analyte itself is more stable. So which diagnostic strategies hold promise, and for which communities? As we said, there is no one strategy that is going to fit all communities affected with HCV. So before we address that, I'd like to just go back and take a bigger picture view as to remind ourselves of what the models of diagnostic care are. So we have the centralised setting, and that is where clinics or smaller labs send their samples to a centralised lab for testing. Then we have the decentralised model of care, in which you have the community centres, sexual health clinics, drug and alcohol settings, where you can receive testing and treatment within those community centres. Centralised testing does come with significant advantages, particularly where you have the ability to have them in developed countries, all the advantages that come with automation, batch testing, reduced operator time. Quality assurance programs are strong in place, and you can easily integrate all your results with existing electronic records. Disadvantages are, as previously discussed, you require highly trained staff, facilities, equipment. But this particularly important is the time, the sample transport time, processing time, and therefore an increased time to result, and therefore a loss to patients and linkage to care. So decentralised models do have many applications and may be amenable to some of the key affected communities for hepatitis C, with reduced time to result, increased linkage to care. But as we were saying before, for many applications are suitable for remote testing and resource-limited settings. So HCV RNA DBS testing is one avenue that holds a lot of promise for hepatitis C. I do apologise for all the acronyms in the title there. But DBS RNA detection, quantitation and genotyping, there are a number of studies that are looking at the sensitivity and specificity of these assays. And overall, they show a strong correlation with plasma, a lower limit or higher limit of detection, about 150 to 250 international units per mil. But considering most hepatitis C patients have higher viral loads than 1,000, this may not be a large issue depending on the question that you're asking. However, there are gaps in the literature regarding dry blood spot RNA testing, and that relates to how well the RNA testing will perform in the real world. For example, in the majority of settings, dry blood spots are likely to be collected under hot, humid conditions and stored for a while in these conditions before getting sent to a central lab. So some of the data looking at stability addresses 4 degrees room temperature, -20, -80, but we really still need to see how the RNA testing performs in the real world. So the clinical impact of testing, again, there are actually few studies that have assessed DBS as an intervention, and quantitatively looked at the impact of DBS testing on rate of testing and diagnosis of Hep C. There are a number of studies, predominantly performed in the UK, in settings such as the prisons, needle and syringe programs, and drug and alcohol settings. And the largest study, a step wedge designed, did actually show that there was no effect of dry blood spot testing, although there were several limitations acknowledged in this manuscript. All the other studies to date show that dry blood spots effectively increase the rate of diagnosis and testing, and are particularly suitable for among people who inject drugs who have a greater acceptability of the dry blood spot compared to venipuncture. So a perfect example of this, and the leaders of this field, are dry blood spot testing implementation in specialist drug services. And it's a large body of work that has been conducted by the Scots, led by Sharon Hutchinson and David Goldberg. And it's been performed in conjunction with the Scots Action Plan, which has been in various phases of development for about 7 years. So they implemented dry blood spot testing in specialist drug services in 2009 and showed it was responsible for the largest increase in the rate of testing, diagnosis and detection of new cases of Hep C, and also the linkage to care. So for them it's been an extremely effective strategy to increase the testing in Scotland. Another application that is available but has not yet been implemented or tested thoroughly at all for hepatitis C is the concept of online self-collected DBS, which is something that we're particularly interested in. So by this, I'm referring to someone who doesn't necessarily feel like they're able to be engaged with the standard or traditional care settings, getting access to a dry blood spot kit. This might be through ordering online, or it might be through peer worker strategies or deliveries. Deliver the kit, you have self-sample collection, you do a finger prick, put some drops on the paper, let it dry, and then send it back in the mail to get tested and referral to care as required. As I was saying, there are no tests specifically studies looking at this for HCV, but for HIV, they've done, in conjunction with the Dean Street Express Clinic in London and the Terence Higgins Trust, where they've assessed self-ordered online dry blood spot testing for HIV in comparison to testing through the sexual health clinic. And they found equal results in terms of recruitment rates and testing results, but they found that dry blood spots were able to cover a broader geographic area and reach others that may not have been able to access the clinic in London, and also diagnosed higher numbers in specific key populations, such as in the example they gave, African American heterosexual men were more likely to go and get tested or get a result compared to going into the clinic. So point of care testing that we've been discussing, there are a number of studies obviously looking at rapid testing for antibodies, but there are very few studies or no studies that actually look at testing active infection on site. So what we need to do is a large number of studies to get the data to support this. We not only have to include the accuracy and clinical point of care testing, but we have to assess the impact of this intervention on the rate of diagnosis, time to treat, and linkage to care. And then assess the differences in cost and cost effectiveness overall to help the public health intervention. So point of care and mobile medical clinics is obviously another avenue that all of these testing facilities may be implemented into. So one example of this was in New Haven, and they recruited, of all the people, 134, so over 1,000 people that came to the clinic, they offered testing. They offered point of care testing or standard, and it was roughly equal, which actually surprised me. But of the people that accepted point of care testing, 94% of those were linked to care, and only 18% were linked to care if they had the standard of care testing. As a result, probably a direct result of the instant result engagement, talking to the patient and then referring them to care immediately rather than having to wait for results and come back for Sorry, this doesn't... Thank you. So, of course, there are many examples. Here's an example in Philadelphia, the onething.com, where mobile clinics provide free HCV testing and HIV testing, and there are examples of this also in France. But we have to think about how this free testing is integrated with not only treatment, but all the other interventions and counselling services and education about preventing the risk of reinfection. There is an example that Bob probably is more familiar with this than myself about clinics, mobile medical centres run in Ukraine, which offer free, interferon-free DAA treatment to all those that test Hep C positive at their mobile centre. So core antigen testing is going to be discussed by Leah in her study after this, so I'm not going to go into too much detail, but just to provide the background, the core antigen has been renewed interest, and overall they show that there's a good correlation in plasma compared to RNA testing. And although not many studies have been done recently, the earlier studies did show there is potential for this to help monitoring treatment response. A recent study in the last couple of weeks has been published looking at the application of core antigen testing to dry blood spots. And this study is indicating that while the specificity is perfect, the sensitivity is reduced, so we still have to explore this further as to whether it can be applied. So this is something that has been discussed. So in conjunction with the simplified treatment that we have, a question from the audience was discussing how is this going to be the follow-on treatment? How can we scale up therapy in the context of the new DAA treatment? So currently, as you can see, with the current therapies, in addition to all of the bloods, prior to treatment you need to get your antibody tested, your genotype testing done, quantitative RNA, and your liver fibrosis assessed. Following treatment, then it's recommended you have viral load tested at week 4, end of treatment, SVR12 and SVR24 if required. The hope and prediction is that with the new therapies, they will be able to be scaled up also because of the simplified diagnostics, the reduced levels of toxicity, and the simplified treatment, pan-genotypic strategy, will require fewer diagnostic tests. So it's predicted that a qualitative RNA, you just need to know, or a core antigen, just need to know if the patient has Hep C or not, and assess their fibrosis prior to treatment. Then following treatment, you may just need one test at SVR12 to determine whether they have cleared or not. And it's predicted, some people are suggesting it may not even be cost effective to do that follow-up qualitative test. It might be similar to antibiotics in the future where you're infected, you get your treatment, and then you walk away. You assume that the majority of patients are going to respond. And there's the application of self-collected dry blood spots as a tool to help this monitoring process. So the patient may receive a kit, take it home, and at a week SVR12, get a reminder to send their result in to confirm a cure. Next slide, please. Thank you. So the steps toward implementation there are many, of course, and we're really at the stage of understanding the clinical sensitivity and specificity, but we need more studies with regards to core antigen testing, rapid diagnostic tests for RNA, to determine the clinical impact and the costs, evaluate the costs. Once you have all this data, of course, there's a massive amount of modelling and evaluation to determine whether the companies who are developing the cases can actually put together a business case to convince that it's worthwhile going towards the next stage of getting submissions and regulatory approval, because there's obviously timing and cost implications involved. And then finally, government negotiations, reimbursement and implementation rollout are down the track also. So we're still at the very early stages of this, but we're collectively gathering all the data required, and that simply holds great promise for hepatitis C. So in summary, hepatitis C diagnostic simplified strategies are likely to help close some of the gaps. Promising strategies include centralised dry blood spot RNA testing, centralised core antigen testing in plasma, portable care devices for active infection, self-collected DBS for diagnosis and treatment, And obviously we need a huge coordinated effort from everyone involved, the affected communities, public, private, NGO ventures to make this happen. So I would like to acknowledge my team back in Australia, led by Professor Greg Dorr, and the teams that I work closely with at the Kirby Institute and the hospitals we're associated with, and of course the funding. So thank you for listening, and I'm happy to take questions. Thank you.